My name is Ben Martin. I am, along with being a co-lead of the early stage researcher working group in OHDSI, I'm a research associate at Johns Hopkins in the section of biomedical informatics and data science. And along with my co-authors, Will Kelly and Christopher Meckley, also at Johns Hopkins in the Department of Rheumatology, I'm here to talk about our paper titled Identification of Adult Dermatomyositis Patients Using Real-World Data Sources. And for the informatics clinical translation of nomenclature here, we did use the term computable phenotype, which is what I understand this to be all about, but it was suggested by the editors of the journal, which is a clinical journal, the Arthritis Care and Research Journal from the American College of Rheumatology, that we use the term case identification algorithm. So if you see that, that just means computable phenotype if you're a computer person. So our paper just got published in August of this year. The main objective, and this is part of a larger research series of research endeavors we have to use large-scale harmonized data to answer clinical questions and extend guideline-driven evidence for rheumatological disorders, but specifically for some of the more rare forms like dermatomyositis, which has an incidence rate of 0.2 to 2 people per 100,000 person years, or that's 2 to 20 per 1 million person years. And then the prevalence worldwide is estimated 2 to 25 people per 100,000 people. So the key rationale here is that OHDSI and distributed large-scale harmonized data networks provide not only an opportunity, but kind of the only way forward for us to answer some clinical questions using observational data in rare diseases. So the other thing I want to mention is this particular part of our endeavor, this paper, this project, heavily leaned on the OHDSI tools Atlas, Cohort Diagnostics, and Phevaluator. So, you know, the shout outs go to Chris Knoll and Frank DeFalco and Gautam Rao and Azza Shobi and Jill Swirtle and many other people who I'm sure helped to develop and maintain those tools. The main thing though, Phevaluator, when this part of our project started, was kind of a new tool. And so this was a really big opportunity to do something cool, which was provide the case identification algorithm metrics to evaluate how well they perform across databases using Phevaluator. So we could produce those sensitivity, specificity, positive predictive value, negative predictive value, typical 2x2 matrix or confusion matrix metrics across all of the databases that we tested the computable phenotypes on. So this is something that has multiple papers to explain this in more detail. Thankfully, we had Joel to help us along the way, but the big picture is that we use particular phenotypes to train, or cohort definitions to train a patient-level predictive model solely for the purpose of then evaluating the actual phenotypes we want to test for use in a research question. So we tested these phenotypes on 11 databases, Johns Hopkins, Stanford, and Columbia, and then the usual lineup of databases that the J&J folks have access to and are harmonized to OMOPS. That's the Japanese Medical Data Center. I forget what that JMDC acronym stands for, but the market scan databases, Optum's databases, IQVIA, and then HealthFarity as well. And this is the main result. So there's another cool result that we didn't really think about early on, but realized we had an opportunity to get to, which is even cooler than our main result. But this is what we came for. We wanted to test, we tested 8 different algorithms, each with like iterative criteria that we thought would help us define what is that ideal balance between sensitivity and specificity, or sensitivity and positive predictive value. So the whole art of phenotyping here is how much criteria do we need to make sure that we are including all the patients we want to include and not including the patients we don't want to include, while also not excluding the patients we want to include and including the patients we don't want to include. And if I didn't say that right, you know what I'm getting at with that statement. So the balance between true positives, false negatives, and true negatives. And we also wanted to have an algorithm that could identify patients with dermatomyositis in terms of prevalence and incidence. So there was two that we picked out as having the, compared to the others, and the 8 different ways we could define a patient and identify a patient with dermatomyositis. The prevalent and incident phenotypes, the only difference there were that the incident one had a 365-day minimum observation period prior to the index date. So that's increasing the likelihood that when we do pick up on dermatomyositis, it's also the doctors at that health system picking up on dermatomyositis for the first time in that patient's trajectory. And then the prevalent phenotype did not require any prior observation. So they could have already been an established patient with dermatomyositis, but we would pick up on them no matter where they're at in their DM etiology journey. Okay, so this was the very cool result that we didn't realize we were going to get to do, but we did. We had the Myositis Center had already done some very rigorous gold standard manual chart review for dermatomyositis and other idiopathic myopathies. And so this was already there. We didn't have to go and do this. The work had been done. All we had to do was connect the patients that we identified from our OMOP cohort definition and cross-reference those with the manual chart review. And this isn't just folks who have a clinical training reviewing EPIC. The Myositis Center team actually followed a very rigorous series of classification criteria for specifically for performing gold standard review of these certain idiopathic inflammatory myopathies. So this was already in existence. We were just able to connect it and provide what was a novel, well, we hoped. There was a funny moment where we were producing these results and we were realizing that depending on how this goes, our whole paper and everything we talked about doing henceforward was on the line here. But thankfully it came out that pHeValuator actually does a very strong job of estimating the performance of the evaluation of phenotype algorithms. So we compared the calculated sensitivity for our phenotype algorithms from the rigorous manual chart review and pHeValuator, and the metrics were very close for at the case identification algorithm level, and then also overall with a kappa agreement of 0.7, which represents substantial agreement. And the other cool thing was that this was at the patient level. So my understanding of the pHeValuator validation to date is that it is comparing calculation of these metrics at the aggregated metric level. So this was actually using individual patients who were able to be confirmed as either a true positive, false positive, or true negative or false negative. And it was also done for the same patients in the same database at the same health system, and it came out to be very aligned pHeValuator with gold standard review. So that was very encouraging, and we were excited to publish this evidence that pHeValuator seems to be a valid tool for evaluating phenotype algorithms at scale. And on that note, want to give a big shout out to Joel Swirtle, who helped us tremendously in mainly understanding how this tool works, opening up the code, understanding every parameter, sending our results to him telling us what we should do next. So I wanted to put him on a big gold star, because we couldn't have done this particular part of our work without him and his patients. So please do go check out his tool at github.com OHDSI/pHeValuator. And then of course some other key collaborators that I have to shout out, being Gautam Rao and Azza Shaobi in the OHDSI phenotyping working group, who also ran our cohort definitions on the J&J databases, and then Hannah Morgan Cooper at Stanford and Thomas Falconer at Columbia for running our package and working with us through that process. So thanks, and also to our other co-authors and everyone else who helped us on this project. So thank you, and thank you for letting me share our work.